Hello everyone, today I am going to teach you about purine salvage pathways and related disorders. So, purines are actually synthesized in two ways. One is called de novo synthesis and another one is salvage pathway. So, de novo synthesis is nothing but synthesis of purines and pyrimidine bases from amino acids. So, the amino acids can be anything. The main amino acids that are used are glutamine, aspartate, glycine, etc. So, salvage pathway actually involves conversion of nucleotides to free bases and again conversion of free bases to their respective nucleotides. So, the nucleotides like GMP, IMP, and AMP are converted to their respective free bases like guanine, hypoxanthine, adenine, and then they are recycled back to nucleotides and they are incorporated to the DNA. So, how does this process take place? So GMP and IMP, as you can see, both of them are converted to xanthine, hypoxanthine, and GMP can be separately converted to guanine. Okay, and these hypoxanthine and guanine are the free bases. Okay, so these free bases are actually converted back to GMP and IMP with the help of HGPRT, which stands for hypoxanthine guanine phosphoribosyltransferase transferase. So they use PRPP. PRPP is synthesized from ribose 5-phosphate, as you can see over here. So they use the PRPP and they are converted back to the respective nucleotides. Okay, and some of the hypoxanthine is actually converted to xanthine, then to uric acid, and is excreted out with the urine. So here hypoxanthine to xanthine and xanthine to uric acid is converted with the help of an enzyme called xanthine oxidase. So this xanthine oxidase converts the free bases to uric acid, which gets excreted, and some of the free bases are then again converted back to nucleotides. So it's like a continuous recycling process. Now coming to AMP, AMP is actually converted to adenosine, which is a nucleoside form, and it cannot be converted to adenine, as you can see over here. It is converted to inosine and then incorporated into this pathway to form hypoxanthine, xanthine, uric acid, or this hypoxanthine is again converted to IMP and this IMP is converted to AMP. So the formation of AMP is through the formation of IMP first. So this adenosine converting to inosine is mediated by an enzyme called adenosine deaminase, and conversion of guanine and hypoxanthine to their respective nucleotides is mediated through an enzyme called hypoxanthine guanine phosphoribosyltransferase transferase. So these two are the most important enzymes, and the disorders I'm going to talk later are related to the deficiency of these enzymes. So this is the salvage pathway which occurs in our body continuously. Okay, so as you can see, the GMP, the IMP, and the AMP are actually synthesized de novo first. So they're synthesized de novo and they are converted to free bases and they are salvaged again to form the nucleotides. So the de novo synthesis and the salvage pathway, both of them occur simultaneously in our body. Okay, now let's go to the purine salvage deficiencies. So coming to purine salvage deficiencies, the first one is called adenosine deaminase deficiency. So as you can see, we know that adenosine deaminase is required for the degradation of AMP and adenosine to xanthine, and also salvage of adenosine back to AMP and to ATP. Okay, so in adenosine deaminase deficiency, because there is deficiency of adenosine deaminase, the adenosine cannot be degraded, which will increase the AMP, okay, and which will increase the DATP because they cannot be degraded. So the DATP increase in DATP is actually noticed by the ribonucleotide reductase. This ribonucleotide reductase, as you know, it is an enzyme in de novo synthesis. Okay, so this ribonucleotide reductase thinks that okay, there are a lot of nucleotides already present, so there is no need to form new nucleotides on new nitrogen bases. So the ribonucleotide reductase activity will decrease. This will decrease the synthesis of new bases, which will eventually decrease the synthesis of DNA, right? So the ADA deficiency is mainly seen in lymphocytes, because lymphocytes are actively replicating cells. They need to replicate every day. So for replication, you know that DNA is required. So because there is decreased synthesis of DNA in ADA deficiency, mainly the lymphocytes will get affected. If new lymphocytes are not formed, there will be deficiency of lymphocytes. Okay, so that leads to a condition called SCID, which stands for severe combined immunodeficiency, where there is deficiency of both B lymphocytes and T lymphocytes. So adenosine deaminase deficiency is one of the major causes of severe combined immunodeficiency. Okay, let us talk about Lesch-Nyhan syndrome. Lesch-Nyhan syndrome is because of deficiency of HGPRT. So we know that HGPRT converts guanine and hypoxanthine, which are free bases, to the respective nucleotides, that is GMP and IMP, right? If there is deficiency of HGPRT, there will be decreased conversion of this guanine and hypoxanthine to their nucleotides, which is nothing but there is deficiency of salvage pathway, right? So if there is deficiency of salvage pathway, to compensate for the deficiency of nucleotides, the de novo synthesis will increase due to increase in PRPP aminotransferase activity. So PRPP aminotransferase is an enzyme in the de novo synthesis. The activity will get increased and there is increase in de novo purine synthesis. So because there is increase in de novo purine synthesis, there is increase in hypoxanthine formation, but the hypoxanthine cannot be converted back to IMP or GMP or AMP because of deficiency of HGPRT, right? So the hypoxanthine will be converted to xanthine and then into uric acid, and which will be excreted. So indirectly there is increase in uric acid production, right? So the uric acid levels in the body will increase, which will lead to the specific symptoms over here. So Lesch-Nyhan syndrome actually causes hyperuricemia. So this hyperuricemia is seen as sodium urate crystals in the urine, and also deposition in the joints will lead to gout, okay? And this will also lead to self-mutilation and aggression. So the uric acid will go and deposit in specific areas in the brain, which will lead to self-mutilation and aggression, okay? So there is no clear-cut physiology why self-mutilation occurs, but it is very specific for Lesch-Nyhan syndrome. And also there is another diagnosis you see self-mutilation, that is Fragile X syndrome. So both Lesch-Nyhan syndrome and Fragile X syndrome cause self-mutilation, aggression, dystonia because of deposition of this uric acid in the muscles. There will be tension in the muscles. Okay, these are the main symptoms for Lesch-Nyhan syndrome. So first side has a very good mnemonic, HGPRT. So H stands for hyperuricemia, G for gout, P for pissed off, that is aggression, self-mutilation, R for red orange crystals in the urine, which are nothing but sodium urate crystals, and tense muscles, that is dystonia. So treatment is through allopurinol and febuxostat, which will decrease the formation of uric acid from hypoxanthine. So they will directly inhibit the xanthine oxidase and they will decrease the formation of uric acid. Thank you.